How's it going, class? This is Mr. Miles coming to the podcast, and today what we're going to talk about is we're going to talk about viruses. Okay, so viruses. These guys are interesting, guys, because they don't fit into any specific category of living organisms. As you know, we have like domain, kingdom, phylum, class, order, family, genus, and species. So with the domain, we have three. We have the bacteria, we have the archaea, and of course we'll have the eukaryotes, and under eukaryotes we're going to have four different types. So the protist, the fungus, the plants, and animals. Now, if you notice, viruses don't fit really anywhere into here. They'd be outside of this because this is dealing just with living things. Viruses are not alive. They have certain characteristics that would make them seem like they were. So we got to remember that viruses, they have structure. They're able to reproduce, but only when they're inside of a host cell. And we'll talk about that. They have genetic code, so they can have DNA or RNA, and most of them are going to have RNA. They don't grow and develop. They don't respond to the environment, and they do change over time. But that's not enough to qualify as living life. So let's talk about what exactly a virus is. So a virus is a parasite. And we call it a parasite because as a parasite, it needs to infect a host. Now, simply what a virus is, if we're just looking at the structure, it's going to have an outer covering, which we're going to call a capsid. So, if you have the influenza virus, you can look it up. It just looks like a ball with these, almost look like suckers coming off the top of it. So, there's hairs with little discs that look like suckers attached to the end of it. And so that'd be the influenza virus. You have the bacteriophage, which is really cool because it just simply means the bacteria eater. So let's draw this. And then kind of like that. There you go. So they have this capsid, and what it is a protein coat. And as a protein coat, it helps protect what's inside of it. Now, if you were to look inside of it, about 70% of these guys are going to have RNA. Some of these guys are going to have DNA, and if you remember, that's that whole A's, T's, C's, and G's. If it's DNA, it would be A, U, C, G. If it was RNA. So here's RNA, here's DNA. Okay, remember A goes to T, C goes to G, and in RNA, A goes to U, and C goes to G. So anyway, so what you have here is you have the protein coat. Inside you're going to have its genetic material. And they're going to be parasite-specific hosts, meaning they can only replicate whenever they use whenever they use a host. And most of the time what happens in these guys is they are going to damage their host, and so that's why you consider them parasites. They have a relationship with the host, but it's not a good one, so it's a parasitic type of relationship. So what will happen is, here we have our happy cell. So he's just minding his own business, and so let's say this is a lysosome right there, but it looks like a smiley face. And there's our nucleus, and inside the nucleus we know that the cell has DNA. Well, what a virus will do is, if a virus is seen out here, and if you notice I drew the virus very small, because the virus is much, much smaller than a cell. Okay, sometimes the cell is going to be 400 times, not 400, it's going to be 100 times bigger than the virus is. And that's because what the virus is trying to do is it's trying to get inside of the cell. Now, these protein coats are really important because on the protein coats, remember I put out these little suckers that are on the influenza virus, which cause like Spanish flu and colds and stuff like that. These things are called protein keys, and these keys allow it to get inside of a cell. If you remember, a cell will have a layer around it called the cell membrane. So there's our cell membrane, and so our cell membrane keeps certain things out and lets certain things in, lets certain things out, doesn't let everything out. Now, a virus will try and mimic something that would go inside of a cell, so it's going to use this protein key. It'll come up to the edge of the cell, and if it matches, it can then go inside of the cell. Now, this is why not everything can get you sick. Sometimes if somebody's sick around you and you're around them a lot, but you end up not getting sick, it could be that your body is looking for a different specific protein key. That's also like why when you have the flu, you'll get really bad sinuses and aches up there, but other places won't really be affected. That's because all of your cell membranes are a little bit different, depending on what type of tissue it is. So if it's a little bit different, these protein keys won't work, but influenza, if it works, it'll go into your sinuses and it'll infect you and you will not feel good. So the way it does that is it goes inside of the cell. Once inside, it wants to get to the nucleus. Okay, it'll get brought to the nucleus because what the cell membrane is doing is it says, oh, the protein keys match. This should be here. Let's bring it to the nucleus. And so the cell gets a nice little ride up to the DNA inside of there. And then what it'll do is it'll take the DNA. So here's our DNA. So there's our, well, crap, let's crap the DNA. Let's try again. So there's our DNA, all ugly. And again, remember, this is in a double helix pattern. It'll go and it'll insert its RNA or DNA. It's just terrible genetic information. It'll stick it inside of the cell. Once inside of there, your cell can't tell the difference between that and the regular DNA that was already there. So the mRNA will come in, it'll copy down any information it has in there, and if a virus went ahead and infected it, it will take that information. So let's say there's a new information. The mRNA will take it out to the ribosomes, and the ribosomes are the guys that make the proteins. But this time what it's doing is it'll simply make a virus. So those instructions are for a virus. And this happens many, 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 many times. And what can happen over time is the viruses grow in such large number that they will simply lyse the cell. And what it means to, And what it means to lyse the cell is it means to break it open. Okay, so these things are being tricked, and then the cell will simply rupture and outpours all of these new viruses, and they can go and do that other thing. So let's just do a little recap of what just happened here. So a virus tricks the cell membrane into letting it in because it has the matching protein keys on the outer coat of the virus. It'll then go up to the nucleus, be brought inside of it. It'll insert the genetic information that was protected by the protein coat. It'll insert into the DNA. The DNA will just be doing copying like it normally does. The mRNA will also be coming in to make proteins like it normally does, but when it reads that part of the DNA, unfortunately, it's going to give bad instructions to the ribosomes to make a lot of viruses, and then it can lyse out and make more. Now, there's two different ways that they can infect. The way that we saw is a lytic infection. And so a lytic infection, what that is that's where it's just going to go right in and trick the cell into making more of it. Now, a lysogenic, so lysogenic infection, what this is going to do is instead of doing what we just saw right there, it'll go and it'll hide inside of the DNA. So it hides inside of there, and if you remember, when mitosis is occurring, what's going to happen in interphase is it's going to double the DNA that it's making. So normal mitosis occurs, and now if that viral DNA is there, it's going to multiply, and then it'll multiply again, and it'll multiply again, and it'll multiply again. So eventually you're going to have a whole bunch of cells that have that viral DNA, and after a particular cue, all the viruses will start being made, and it'll get pumped out into the body. So a lysogenic one is where it kind of waits, whereas a lytic one, it just kind of starts making itself almost immediately. You can see a huge advantage to this because you can hide inside of the DNA without the infection ever really showing up, or without you even getting sick, so you can still be a transmitter without realizing it. Whereas in this one, you're going to start getting sick pretty fast as it starts breaking apart different cells and entering into new ones. And so it's a little bit harder to transmit because when you see a guy sneezing, you're going to try to stay away from him. Whereas this one, while it's waiting for the cell division, sometimes it can wait a long time. HIV, this one can stay dormant for about 10 years. So it waits for 10 years worth of cell divisions before it finally starts making more and more of its viral tissue, enough of it for you to notice. So unfortunately, within that 10 years, a lot of people can be going around spreading it. And so, yeah, that's the main things about viruses. I guess I don't really have too much else. That's just a brief overview. So remember, they're not living. They need to use a host in order to spread themselves around. They're going to do that by tricking the cell into making more of it. And then there's two different types. Lytic, which can start breaking open and dividing really rapidly, making more of itself, whereas lysogenic is where it's going to hide inside of the cell divisions and just keep multiplying itself until it's ready to go and infect everything. So there's your podcast. If you guys have any comments or questions, just feel free to ask me in class or shoot me an email. All right, take care. Bye.